Welcome to this advanced module on molecular logistics. We've previously explored the basic walking of the kinesin protein, but today we're peeling back the layers to analyze the complex biophysics, regulatory coordination, and clinical breakthroughs in neural transport. In our previous sessions, we established the microtubule network as the cellular highway. Today, we focus on the drivers. We'll dive into the Brownian ratchet model, explore the mechanical zipping of the neck linker, and look at how we are using AI and gene therapy to fix transport failure in diseases like CMT2A and KAND. By the end of this 10-minute deep dive, you'll be able to analyze the stochastic physics of molecular motors, master the leverage mechanics of the walking cycle, and evaluate the specific genetic interventions currently in human clinical trials. To appreciate why these motors exist, we have to look at the math of diffusion. In a small cell, cargo can drift. But in a neuron with a meter-long axon, the time required for a protein to diffuse from the spine to the toe isn't measured in hours or days. It's measured in centuries. Active transport isn't just an upgrade. It's a mathematical necessity for life. Because diffusion time scales with the square of the distance, random motion fails at the scale of a human body. Kinesin bypasses this limit by converting chemical energy into directional work, moving at speeds that allow a neurotransmitter to make the journey in just a few days of continuous walking. Inside that axon, kinesin lives in a world of thermal noise, a constant random bombardment by water molecules. It doesn't drive forward like a car on a road. Instead, it functions as a Brownian ratchet, turning random chaos into focused progress. This is the Brownian ratchet model. Kinesin uses random thermal energy to explore its surroundings. When the neck linker docks, it creates a mechanical bias. This bias doesn't force motion. It simply prevents the motor from diffusing backward, effectively ratcheting random vibration into unidirectional progress. The mechanical force for each step comes from a tiny segment of 15 amino acids called the neck linker. When ATP binds to the lead motor head, this segment zips onto the motor domain. This structural change acts like a spring-loaded lever, throwing the rear head forward toward its next binding site. This zipping mechanism throws the detached head 16 nanometers forward to land 8 nanometers ahead of its partner. It is a masterpiece of molecular engineering, leveraging chemical energy into precise mechanical work with near-perfect efficiency at the nanometer scale. Speaking of efficiency, consider the energetic cost. Kinesin consumes 1 molecule of ATP for every single step. In a high-friction, crowded environment, it manages to convert that energy into movement with an efficiency that puts man-made internal combustion engines to shame. It is truly the cell's most efficient machine. The hydrolysis of ATP resets the motor, allowing the neck linker to unzip and prepare for the next thermal search and docking cycle. This continuous loop of energy to work is what keeps the cell's logistics running 24 hours a day throughout your entire life. Next, we look at the cargo. Vesicles aren't rigid crates, they are fluid, lipid bilayers. Research now shows that kinesin motors aren't fixed in place. They can diffuse freely on the surface of the cargo, allowing them to cluster dynamically where the load is heaviest or where obstacles are detected. Binding kinetics are the primary bottleneck. The rate at which a motor reattaches, the K-on rate, determines the total run length. For a typical neuron, it takes a cluster of approximately 35 kinesins working in sequence to ensure the cargo doesn't detach before reaching the distant synapse. The highway itself isn't passive. Microtubule-associated proteins, or MAPs, act as the cell's traffic controllers. Proteins like MAP7 can increase the binding rate of kinesin by nearly tenfold, essentially greasing the wheels for transport in specific directions based on the cell's immediate needs. By regulating where MAPs attach to the microtubule, the cell can create preferential fast lanes for vital cargo like mitochondria, ensuring they reach energy-hungry zones, while waste products are efficiently recycled back to the cell body for processing. But what happens when two different motors pull in opposite directions? This leads us to the molecular tug-of-war. A single vesicle often carries both kinesin, pulling out to the synapse, and dynin, pulling in to the cell body. It is a literal mechanical struggle for the soul of the cargo. The forces are remarkably asymmetrical. A single kinesin motor is 6 times stronger than a mammalian dynin. To reverse direction, the cell must either cluster many dynins together to win the tug-of-war, or use a molecular switch to turn the kinesins off entirely. This brings us to the paradox of codependence. In many experiments, if you inhibit one motor, transport stops in both directions. This proves the motors aren't just fighting, they are coordinated through adapter proteins that act as intelligent molecular switches. Adapters like JIP1 serve as the cellular GPS. They can auto-inhibit kinesin until it reaches its specific destination or relieve that inhibition to trigger a run. This coordination ensures that the internal logistics remain efficient rather than a chaotic, wasteful struggle for control. We see the human stakes of this coordination in the clinic. Charcot-Marie-Tooth disease type 2A or CMT2A is the quintessential transportopathy. Caused by mutations in mitofusin 2, it leads to the slow starvation of the long nerves in our legs, resulting in progressive muscle weakness and sensory loss. In these patients, even a single amino acid change in the motor domain can disrupt the ability to bind cargo or walk efficiently. Supplies of energy cannot reach the distal synapse. The neuron begins to degenerate from the tip backward, a process we are now learning to reverse through genetic engineering. A closely related spectrum is KAND, KIF1A-associated neurological disorder. Here the mutations affect the fastest motor of the family. Without a functional KIF1A, synaptic vesicles containing vital neurotransmitters simply can't be delivered at the speed life requires, leading to developmental delays and seizures. By mapping these genetic mutation hotspots, we've identified how the motor core can be stalled. But this precision also gives us a clear target. If we know the exact structural failure, we can design a specific genetic fix to restore the motor's mechanical purpose. This brings us to the most exciting breakthrough in modern neurology, antisense oligonucleotide therapy, or ASOs. We are now in clinical trials using ASOs to silence the mutant copy of a gene, allowing the body's own healthy copy to restore the proper transport balance to the cell. In pioneering clinical cases, patients have shown remarkable functional improvement after targeted ASO treatment. By restoring the cluster of healthy motors, we can clear the axonal traffic jams and stop neurodegeneration in its tracks. We are moving from observing the failure to editing the cure. To track this, we are using AI to identify cellular fingerprints of disease. By analyzing pattern recognition and mitochondrial damage across hundreds of different mutations, we can finally measure exactly how well a therapy is working at the microscopic level before symptoms even improve. These AI biomarkers allow clinicians to tailor gene therapy to the individual, ensuring that the molecular fingerprint of the disease is being successfully erased and the logistics of the neuron are being fully restored. It is the dawn of truly personalized molecular medicine. Finally, consider the sheer magnitude of this effort. If a kinesin motor were the size of a human, it would be walking at 600 miles per hour while carrying a backpack the size of a large building. This is the level of effort happening inside you every single second. This human-scale perspective highlights why these proteins are the ultimate symbols of purpose. They operate in a high-friction, noisy environment and yet achieve a level of directional commitment that sustains life across the vast axonal highways of our nervous system. As we synthesize these concepts, remember, progress is a mechanical commitment. Success comes from the persistent, coordinated effort of the next small step. Whether in biology or in life, the key is to keep the molecular logistics moving forward. No matter the noise, keep walking. To summarize, we've analyzed the physics of stochastic motion, the mechanical leverage of the walking cycle, and the clinical reality of motor failure. We've seen how genetic intervention and AI can restore the logistics of the cell, providing a path forward for modern molecular medicine. In our next module, we'll do a similar deep dive into dynein, exploring the complex architecture of retrograde flow and the molecular tug-of-war. Thank you for joining me for this advanced session. I'll see you in the next lesson.